We will continue on the application side from now, and wherever required we will introduce some bit of theory. Okay. So, then we will start the application part. Okay. So, let us start the application of fluorescence spectroscopy, where we can use this, a profound, extremely powerful technique where we can use it. So, I will give you some example which is from our lab, means not only from my lab, but our colleagues lab, some real life experiment. You know a protein called tubulin? What is interesting property of a tubulin? Where you find tubulin? Microtubule is a polymeric form of tubulin, right? Where do you find tubulin? Okay. And what it does? Shapes, sign, yeah, it helps in cell division, right. So, tubulin is extremely important protein target for cancer therapy. So, there are this you will study in cell biology, that tubulin is an interesting molecule. It can form the dimeric like a dimeric or multimeric assembly. It looks like rod, it looks like a rope, and that attaches to spindle fiber, pulls apart, cell division happens. Now, if you, and that has a interesting property which is called dynamic instability. What is dynamic instability? Microtubule has two ends, one is growing end, one is shrinking end. एक तरफ से बढ़ता है, दूसरे तरफ से झड़ता है, right? That's the interesting property of microtubule. Therefore, all the cancer drugs, many of the cancer drugs targets this microtubule. Can we name a drug that targets the Taxol? Very nice. Anything else? Cancer drug Taxol is a nice drug that targets microtubule. Any other drug? Colchicine. Anything else? Vinca alkaloid, epothelone. There are many drugs. Over the years, people were so fascinated with microtubule that many drugs has come up. Many of them are clinic that are used for this. But so all the drug discovery goes through basic research. You have to first know if the tubulin is making polymer or not making polymer. So, emission spectra of a tubulin, you can plot it like this, right. So, this is our emission spectra. So, anything you have to, because protein does not stay in anything, it has to stay in a certain buffer. So, first you take a buffer spectrum and then you take a emission spectrum of tubulin. So, what we learned here, it is giving somewhere maxima around 335, this is my tubulin spectrum, right. So, you learned about tubulin, its emission spectrum. Now, here is my tubulin that is there, right. So, and there is a ligand that we are adding it. So, tubulin is here and then we are adding a ligand. Why we want to add a ligand? As I said, all the cancer drugs are targeted towards tubulin. So, tubulin, it is a monomeric unit, its polymeric unit is microtubule. So, if you say stop tubulin from forming microtubule, that is your job is done, or you help microtubule to dissociate, your job is done. So, anything plus end binding or minus end binding or destabilizing microtubule, your job is done, because you do not want this guy to help in cell division. If you perturb it, you are inching towards a drug discovery for tubulin. So, here you can use the fluorescence spectroscopy, a quick way to find it out which molecule binds to tubulin and which does not. So, this is the experiment where you have a free tubulin in black and then increasing concentration of your drug, can be any drug, colchicine, vinca alkaloid, epothelone, whatever you name it, and you plot what is happening here, emission is dropping down. So, this spectra says that when this drug binds and the fluorescence is mostly coming from say tryptophan, looking at the emission maxima, let us say that it is changing the conformation of tubulin and that therefore, it is binding. If it does not bind, it does not change. So, here the delta F, when like a free form and bound form is changing. So, you can plot delta F versus ligand concentration, and you can fit in the equation. Here is the protein concentration, ligand concentration Kd. You can fit this equation to find what is the Kd of that, right. So, therefore, a good Kd means the value is lower in micromolar to nanomolar, it is a good ligand that you have found. So, it is a quicker way to find it out which is binder, which is not binder. Not only quicker way, it is a quantitative way to find it out whether it is binder or not binder. So, tubulin is a monomeric protein, it polymerizes, you have to give something like that, it is an active process of polymerization, requires energy. So, we are using monomeric tubulin, and for monomeric tubulin, this is my spectrum, right. This is my spectrum. So, here is my lambda max, which is 335, it is mostly coming from tryptophan. Now, when we started adding drug that you can name it, say for colchicine, let us name it colchicine. Now, emission is decreasing as we are going from 0.5 to 1 to 2 to 5 to 10 to whatever 40. So, emission is dropping. That is saying that my microenvironment of the reporter fluorophore is changing, and that change is giving me emission drop. That is what we are plotting here, delta F. Delta F means like a free tubulin, drug bound tubulin. You are plotting against ligand to find it out what is the Kd of that drug which is binding with tubulin. That was your question?
Sir,
go ahead.
Like in a simple form, if I am having a system and I am having a tubulin which is labeled with fluorescence, whether it is bound or it is not bound, it is still there in the system, I will still get the signal, then how am I plotting it like that?
So, here what is the fluorophore is a intrinsic fluorophore, which is inside the protein, right. So, it is there, the fluorophore which suppose is a tryptophan is there, but what is happening when you are putting the drug, suppose this is my tubulin and here is my drug is coming and binding here. So, for binding this something is changing here, right, near tryptophan fluorescence, and that change is decreasing the fluorescence, and that because protein does not remain same when drug binds, some change is happening. That change we are monitoring whether it is binding to protein or not. So, if suppose this drug bind, right, drug binding, it changes the conformation of the protein. Now, for microtubule to form, you require a certain conformation of the protein. Now, that conformation has been altered. If that conformation has been altered, now microtubule binding capacity is decreased or lost. So, it will not form that rassa kassi wala rassi which is used for tug of war for the like a cell division. So, that is way it is happening. Fluorophore is there, but its microenvironment has changed, that is what is happening here, right. So, that is another drug I just gave you in the previous, a colchicine. Suppose you are coming up with a new drug, CXI benzo 84, it is a new drug, abhi market mein nahi hai, we are suppose want to bring it, bound to tubulin at a single site. So, essentially you can do all these experiments where fluorescence intensity is dropping in presence of increased concentration of tubulin. Now, in B you can plot it, the CX benzo bound to tubulin in a concentration dependent manner, like you can change the tubulin and find it out. Now, bound and free fraction of the drug is calculated, and you can also find it out 684 inhibits the tubulin fluorescence. So, you can do all sorts of experiment. Suppose your drug is a fluorescent, you can also look at how much fluorescence of drug is decreasing, how much fluorescence of tubulin is decreasing, what is the bound fraction, what is the free fraction, how much drug is required, because always you want a drug to be better binder, right. So, all these experiments are possible, and essentially you can calculate where it binds. So, you can do competitive reaction, already you saturate with colchicine, and because if you want to design a new drug, it has to be better than the previous. Me too type to nahi chalega na, already 10 markets my drug hai, you bring 11-1 which is not better, nobody is going to believe you. So, for that you can even find that it binds to tubulin at a colchicine site. So, you know the colchicine site where you can saturate it, let the colchicine be replaced with this drug. So, essentially these kind of fluorescence experiment, same absorption decay, you can plot it, you remember this kind of curve. Enzyme kinetics, competitive, non-competitive, uncompetitive, all kind of reaction you can plot it and find it out the KD, the competition and the site, because we have found that it is binding with colchicine. So, this kind of experiment essentially there are many like a compound 12 binding at colchicine site, right. So, you can find it out what is the KD, how much it is binding and how it is getting dissociated, right. So, the another example that we had discussed about, so that is about tubulin. Similar kind of approach you can do for antibacterial drug, right, antibiotics and all those. So, tubulin is for human cell division, similarly you can do for bacterial. Same kind of protein which is called FTSZ, that is responsible for bacterial cells to shrink and dissociate. If you perturb the dynamics of this protein, you can get a drug binding. So, FTSZ can be done. There is another protein that binds, helps FTSZ, which is called VIMD. In Mycobacterium sigmatis, FTSZ, possible role of VIMD in bacteria. So, there is another associated protein that helps the like a Mycobacterium FTSZ, you can perturb those. So, FTSZ can be target, associating protein can be target, all of these using simple fluorescence spectroscopy you can target it. Extremely simple, but important and powerful technique for starting your drug discovery approach. It does not require too much money, does not require too much time. So, you have to do a fast screening, you can screen, you can have a developed technique for a fast screening, getting a potent drug molecule using fluorescence spectroscopy. Essentially what you need, a fluorophore which emits and you can measure it, a fluorescent like a fluorimeter and a library of compound which you want to detect it. Okay. So, yeah, most of the time I just emphasize upon the only one, mostly I talked about tryptophan, but it is not only that is there. There are so many fluorophores that are there. So, 3 amino acids that we discussed here, that is endogenous fluorophore, tryptophan, tyrosine, phenylalanine. There is a excitation and emission, phenylalanine quantum yield is very low. Then there is a structural protein where you can find it out, collagen, elasticin. Enzyme has a cofactor like FAD, flavin, FADH, NADH, all of them. Vitamins are fluorescent, K, vitamin A, D. Then there is a B6 compounds like a pyridoxine, pyridoxal, all of these are again fluorescent. Lipids you can use it, phospholipids, ceramides, porphyrin are fluorescent. So, all of these, like it can be indigenous fluorophore or amino acid, some protein, enzymes, coenzyme, vitamins, lipid, porphyrin are there. And other than that, if nothing is there, you can use extrinsic fluorophore. Extrinsic fluorophore जो बाहर से attach करते हैं, you have to synthesize that chemically or isolate from somewhere and then attach to your protein. These are called extrinsic fluorophore. Those can be any dye that you synthesize, like cyanine dye, photosynthesizer, molecular marker like a GFP, YFP and all those are extrinsic fluorophore that we can use it. Right. So, intrinsic fluorophore are essentially amino acids, NADH, FADH, pyridoxine phosphate, all of these are intrinsic, and extrinsic can be whatever we attach. So, let us look at application of various biological problem, starting with intrinsic, which is already protein. So, fluorescence is one of them that can detect the conformational change that happens in protein, and that conformational change can happen either intrinsically or can be induced by something which is binding. So, one can see this kind of motion. It can put it probe the property of an active site of an enzyme. You see any active site that you want to say, active site, some reaction is happening, but all the time it is not like this. It closes, and like when reaction has to happen, it takes the substrate in, does the reaction, opens it up, substrate is released. This is the binding site conformation is changing. Any enzyme does like this. For an example, HIV1 protease, do you know? It is a very interesting molecule. It has a flap like this. It does autolysis or something. So, if you just want to inhibit this HIV1 protease, you have to find the pocket or conformational change that is happening. You come at, bring it something here that locks the conformation. So, HIV is a very dangerous drug. आज तक से कोई भी वैक्सीन नहीं है. I don't know how many you know the history of HIV and war against HIV. नहीं पढ़ा है? ये तो पता है ना what is HIV, huh? And how it spreads. That also all of you know, right? It's a interesting case study that, remember Bill Clinton, one day he came and declared war against AIDS or HIV. Vaccine बना लेंगे, आज तक से नहीं बन पाया. So, what is the effective therapy of HIV? Combination, yeah, there are combination of heart therapy that is done by one guy which is sitting next to our institute. His name is Yusuf Hamid Sipla. It is one of the leader in a HIV treatment drug. So, they have come up with many, like a small molecule that reverse transcriptase protease inhibit करता है, but only problem of such kind of drug, you have like one has to be there. जैसे ही बंद किया वायरस अपना सर उठा लेता है and then it starts a problem, right. So, all of these discovery that I am talking about has been done because people understood how the protein in such organism works, conformational dynamics, understanding the conformation, locking that in the conformation that can be proved using the. So, how enzyme, because HIV protease is enzyme, study and denaturation of protein molecule, location of a tryptophan that you can study using this. Now, extrinsic fluorophore, like many of the dye, rhodamine, fluorescent, coumarin, carboxycymene and aromatic hydrocarbons and derivatives, pyrene, anthracene, all these kind of molecules are extrinsic fluorophore that can be attached to a molecule. Another important one, extremely important, probably if you do biophysics experiment sometime, then you will come across this molecule called ANS, 1-anilino-8-naphthalene sulfate ANS. This is a dye. Other are like a cyber green, it is one known compound, fluorodensyl and this lysine, rhodamine sulfonyl, all these are extrinsic fluorophore. Just spend 1-2 minutes in describing you the ANS, why this is important dye, and when you are doing biophysics, why you have to study this. ANS is an interesting dye. It only binds to the exposed hydrophobic surface area or surface of a protein. Exposed hydrophobic surface. In a protein, do you have typically exposed hydrophobic surface? Exposed hydrophobic surface. Typically a folded protein does what? Hydrophobic अंदर, hydrophilic बाहर, because it has to sense water outside. Typically you don't have hydrophobic which is exposed. Patches होते हैं, but majority of hydrophobic नहीं होता है, because hydrophobic don't like water. Water like them, water they don't like water, एक तरफा love है उधर. Hydrophobic surface like water, water like hydrophobic surface, hydrophobic don't like. So, they don't want to see the water, they want to be inside. But in some cases when the hydrophobes comes out, that is a starting point of protein precipitation, aggregation, amyloid formation, all sorts of change. Conformational change when a hydrophobic comes, So, as I said, hydrophobic को पानी पसंद नहीं है, it likes hydrophobic. So, if suppose a protein was very well folded and for some reason the hydrophobes start coming, now what will it will ask its friend, which is other protein, come and protect me, come and protect me, like this, and it will form the amyloid. And you know what is the role of amyloids, it can damage even neurons, right. So, if you want to understand the exposed hydrophobic surface area, ANS is a fantastic dye that only binds to exposed hydrophobic surface area. So, it gives rise high fluorescence, does not bind to hydrophilic, does not fluoresce, gives high fluorescence in water, binds to. Similarly, there are cyber green is used for understanding the protein fluoride. All of these tensile are used as extrinsic fluorophore. Our issue is that in protein we have only one good intrinsic fluorophore if there is no cofactor, only one good fluorophore, which is tryptophan. Suppose I want to understand, which I am going to talk about, how this portion of protein is talking to this portion of a protein. बेचारा एक ट्रिप्टोफैन इधर अकेला बैठा है, राइट, उससे बात करने के लिए कोई नहीं है. I want how these two guys talking to each other, then what we have to do? I have to attach something here, right? I have to attach something here which can talk to this guy when they come together. So, something like this. So, you require an extrinsic fluorophore, right? Extrinsic fluorophore, let them talk sometimes. So, this extrinsic fluorophore can be attached to an amino acid, and then whenever some conformational change happens, they can talk to each other. We will discuss this in detail, but that is the way you can do it. And so some cyber green or FITC or densile are these kind of fluorophore that can be attached to a protein to understand what kind of conformational change happens. They have different excitation and emission wavelength, right. So, extrinsic fluorophore, it determines the say property of a heme binding protein in hemoglobin, determines the conformational change when enzyme is bound to substrate, determines the strandness of a polynucleotide. Many of such biological phenomena you can probe by attaching an extrinsic fluorophore. Main is interesting is motion that happens in a protein or any biomolecule that one can have it. So, again and again I will remind you this Jablonski diagram, because lots of concepts emerge from here. So, Jablonski diagram, I said intrinsic or extrinsic, the common theme is Jablonski diagram, right. Even if I have to design two probe, how far they are, how close they are, you have to have understanding of Jablonski diagram, because your emission should not overlap if I want them to probe to talk. So, and if I want to do dynamics, I have to choose a property that is there, like a fluorescence happening here, phosphorescence happening here. So, the property of a fluorophore and their time scale determines lots of things, and therefore, Jablonski diagram is important. Coming back to quenching, already we have discussed about quenching. What was quenching? जो चुपचाप एनर्जी ले लेता है फ्लोरोफोर का, राइट. फ्लोरोफोर का शक्ति क्षीण करने वाला मॉलिक्यूल को क्वेंचर बोलते हैं, एंड द फेनोमेना विच reduces it's called quenching. So, one of the, it's not like a, if this is not in the fluorophore, but it's extrinsic one. So, external factor with a practical implication in presence of a quencher, it just takes down, and there is a, even the quencher has some role to play in understanding the protein dynamics. It's a molecule that decreases the quantum yield of a fluorophore by non-radiative process, and absorption of a fluorophore is not altered by the presence of, absorption is not altered, just it goes to the excited state, but while coming back this takes the energy, right. So, energy of excited state is transferred to a quencher molecule, and therefore, a quantum yield can happen, the radiation can decrease and all those, and this has again extremely important phenomena, but there are different kind of quenching happens that tells you the fluorophore information. So, there are two kind of quenching, one is called dynamic quenching, another is called static quenching. Dynamic quenching happens because of collision between fluorophore in the excited state and quencher. Let us go back here, Jablonski diagram. We said that even if quencher is there, this process is not affected. In dynamic quenching, what is happening, since fluorescence is coming from here, the quencher is somewhere taking the energy in the excited state. That is a collision happening and takes out the energy from the excited state. Static quenching, it forms a complex with the fluorophore. Fluorophore absorption happened, but what happened, this quencher has formed a complex. Now energy is dissipated and it does not come. So, the complex has a different electronic spectrum because it has formed a complex with a fluorophore, it has a different structure than a free fluorophore, and therefore, when it returns back from excited state to the ground state, it is a non-radiative. So, dynamic quenching is interaction happening in the excited state. Static quenching means it just got and attached to a fluorophore. It absorbs the light, but the quencher has taken out all the energy just because it has formed a complex. If this fluorophore had been free, it would have given us the radiation, but since this complex has formed, takes the energy. So, these two kind of quenching that happens, and the dynamic quenching, the collision wala process, the dynamic quenching results in a equation that is called Stern-Volmer equation. What it says, Stern-Volmer equation, what it says that I is a incidence light by I 0 is a, that light the photons that comes after fluorescence, minus 1 is k q c q t 0. So, k q is a Stern-Volmer constant, concentration and lifetime, right. So, I by I 0 is k a c q. Stern-Volmer relates the degree of quenching that is expressed in this I by I 0 to a concentration of the quencher here, and this is the constant and this is the lifetime of the. So, rate of constant of quencher is k q. So, each quencher has a quenching Stern-Volmer constant. So, it depends upon how much light it takes. Quenching, dynamic quenching, the quencher is interacting with fluorescence excited state and taking out the energy. Now, remember quencher has to interact with the fluorophore, right. अब किस fluorophore से quencher interact करेगा, जो fluorophore जिसका environment free होगा interact करने के लिए, जो fluorophore buried होगा, quencher cannot penetrate to that. The energy of that fluorophore which is buried inside, quencher cannot reach there, right. Quencher cannot reach, quencher is a molecule. Suppose my tryptophan is inside here and quencher is rotating here, can it reach there? उसको time लगेगा या probably शायद नहीं जा पाए. But if my fluorophore is like this and quencher is rotating here, it is a free, it can reach there. So, concept of quenching, you can find it out the exposure level of a fluorophore, whether the fluorophore is free or buried inside, that is what you can find it out. So, I will give you one example which is all from our lab research. This is a protein which is let us forget the not go into detail, protein called CSC4. It is in the yeast nucleosome variants of H3, protein called CSC4. It has an interesting structure, two domain, one is this domain histone like fold, this is a free domain that is called end domain. It has a two tryptophan, one at 7, one at 178. One in the C terminus domain, one at the N terminus domain, right, two tryptophan. Problem is that being an NMR spectroscopy, what is my dream? To solve the structure of this protein using NMR spectroscopy. बट हर बार जो चाहते हैं वो होता नहीं है. क्यों? NMR spectroscopy requires the high concentration of a protein, few hundred micromolar, but this guy was not coming. 10 माइक्रोमोलर के जैसे ही बियॉन्ड करते थे प्रोटीन प्रेसिपिटेट हो जाता था. एंड इफ यू आर ए PhD स्टूडेंट एंड योर सुपरवाइजर इज आस्किंग गेट मी 500 माइक्रोमोलर ऑफ ए प्रोटीन, उसके बियॉन्ड तुम्हारा PhD नहीं होगा. व्हाट योर रेस्पॉन्स इज गोइंग टू बी? सर मैं PhD छोड़ के जा रहा हूँ घर में, आई विल डू समथिंग एल्स बट नॉट PhD, राइट? बट देन देयर इज ऑलवेज स्मार्टर वे टू डील द थिंग्स. You cannot wait that it goes to 500 micromolar, you can probably get some information even with a 5 micromolar, that using fluorescence, right. So, here you will not get whatever you wanted to solve the atomic resolution structure, but you can learn a lot about protein just only using fluorescence. So, here my 2 tryptophans were there, W7, W178. I said 1 tryptophan is excellent, 2 is not so good, because now the signal will come from both of that. So, first thing we do is let the 1 tryptophan be mutated. So, we make it silent. So, now using single point mutation you can create where only W7 is active and 178 mutated to something like leucine or whatever, you can mutate it. In another case you can mutate this one and keep active 178. So, now we have 2 construct. In one construct only W7 is active and in another construct only 178 active. When I do fluorescence of these 2 protein, we know signal is coming from where, right. Now we can do the quenching experiment, right. So, okay, just forget this graph, just look at this. Here is 1 tryptophan, here is another tryptophan, and this is a modeled structure, right. If I show you a structure you will learn about it, but think that I do not know any structure, there is no structure known. So, think about that. Suppose I do quenching experiment of this and this, where is a possibility that more quenching will happen? W7, because it looks higher. So, you can basically do the KQ and you can see marginally this is higher, not too significant, although model structure says that this is a free and this is buried, but our experimental result saying ulta, no, not so ulta, but it says that for their local environment it is not much different, both seems to be more or less reasonably free, because their KQ is what? That Stern-Volmer constant. We did that. Here is the experiment that we did and we find it out using quenching that both of them are not that free. So, just by doing the simple quenching experiment we could find it out the local environment of the 2 tryptophan, how it is buried or not buried, and we got some idea. So, all the model structure there is still no X-ray NMR cryo-EM structure for this protein, but at least whatever was model they said that this remains very free, this is very well structured. At least our data does not seems to be showing that they are very different, there is some difference but not significant difference. So, another quenching. Now, another important protein which is extrinsic fluorophore, we know green fluorescent protein. It was isolated from a jellyfish and it has excitation wavelength 395, emission is wavelength 509, it emits in green, that is why it is called green fluorescent protein. It is a 27 kD protein and its quantum yield is very high, 0.79, 80% quantum yield, right. So, and only for discovering this protein the scientist got the Nobel Prize in chemistry in 2008, right. So, development of green fluorescent protein, that is important because तुम पूरे चूहे को हरा दिखा रहे हो just because of one protein. So, that is a important experiment that was done. Now, where this fluorescence is coming from? So, GFP is a beta barrel protein consisting of 11 beta strand and alpha helix containing covalently bond chromophore, and this chromophore is a HBI, and the beta barrel is nearly perfect of like a 42 angstrom long and 24 angstrom in diameter, and HBI chromophore is formed by like a by cyclization of tripeptide, which is serine, tyrosine and glycine, and it spontaneously folds into polypeptide chain, and this reaction gives you the like a acid base reaction that we can say oxide like a hydrogen addition gives you a molecule which is fluorescence, and this is the basis of protein GFP is your green fluorescent protein. So, what is the green fluorescent protein? In cell biology, you can use this as a reporter gene. So, typically suppose I want to study this protein inside the cell, I just have to attach a green fluorescent protein. जहाँ जहाँ ये जाएगा, यहाँ यहाँ ये भी जाएगा, right? This protein is invisible, but green fluorescent protein is visible. So, it can tell you where my protein of interest went. Microtubule bind किया, right? Inside nucleus गया, inside mitochondria से कर रहा है, ER से कर रहा है, everything you can just track this protein, and that is a fantastic probe that it has, right? So, that green fluorescent protein can be done. You can do fluorescence microscopy, not a spectroscopy. Microscopy inside the cell you are looking at. Most of the time you do not use fluorescent protein, like green fluorescent protein. You can use even FITC that binds to the protein. You can even do the live cell imaging, right? So, live cell imaging time lapse, यहाँ जो next microscopic facility, beautiful setup that we have, you can really look at the slice wise cell में, right? Live cell imaging, how things are happening inside the cell, and GFP internal chrome 4 does not require accessory cofactor gene product or whatever, you can do it. So, now once people found this GFP, so I had one good protein that can go inside the cell and can report me, but इंसानी दिमाग है उतने पर कैसे मानेगा. मैंने सोचा एक और ऐसा प्रोटीन बनाते तो we can study many other reaction, right? So, then engineering started, protein engineering started. Green fluorescent protein था, blue fluorescent बना दो, red fluorescent बना दो, and now you can have more than one probe, and now how the two protein inside the cell goes and starts talking to each other, you can probe it. So, that is the origin of protein engineering, and you have a variant of GFP, blue fluorescent protein, cyan fluorescent protein, green fluorescent protein, yellow fluorescent protein, red fluorescent protein, all of them, and their wavelength is also different, emission wavelength are different, excitation wavelength are different, and essentially you can have many variants that helps you to identify where my protein is going, my protein is talking to which other protein. So, two tag you can attach and look at the reaction happening inside the cell when the two protein talks, right. So, that is a beauty of the engineering that we had, right. So, it all depends upon now what we want to do and how depth we want to go. So, a simple experiment was steady state fluorescence, then we looked at time decay fluorescence, now we are talking about inside cell imaging. So, you see if we want to do more and more, your instrument has to be more sophisticated, more money, more information, more patience. You have to be in dark sitting hours and hours to do all these experiments, right. So, that is what you have to invest in getting such depth knowledge. Now, confocal microscopy is one of the important tool in biophysics, biochemistry, cell biology and all those. All sorts of microscopy is important tool to understand the wonderful phenomena that is happening inside the cell. Otherwise, we had no way that we know what is happening inside the cell. Now, we can send some reporter molecule which can report what is happening inside the cell, right. So, that is all from the fluorescence and its application. The another things probably I have already discussed how the fluorimeter works, but if you are interested briefly we can talk about. That is a recap, the fluorescence coming back to the ground state, Stokes shifts and all those, and this is the detail of the fluorimeter, sample chamber, light source, monochromator, and then emission monochromator and photodetector. That is a simple fluorimeter. Right. So, you can measure the lifetime, you can measure the vibrational relaxation, Stokes shifts and all those, and extremely with simple spectrophotometer. You require a light source, excitation monochromator, the emission monochromator and a detector. That is extremely simple. So, extremely simple fluorimeter. For MSc level this is enough, but when you want to even go more and more, microscopy you have to, concept remains same, you require a light source, but if you want to do fancy experiment, this light source changes to laser source. For photo detector you require a single photon counting instrument, because you want to have a even feeble photons that is coming out from the cell, you want to measure it. So, it depends upon how depth you want to go, you require a different kind of setup to be done, right. So, that is simple experiment. Now, next class onward we will be looking at some of the advanced concept of fluorescence and its application in biology. Right. So, let us stop it here. Thank you very much.